Hello everyone! Thank you very much for the kind introduction and for inviting me to speak today. As mentioned, my name is Jana Seiler and over the next 50 minutes I will discussing genome editing techniques in honeybees, focusing on multi-site image of neuronal activity using genetically encoded calcium sensor in the honeybee. First, let's take a look at the structure of the bee colony. On the one hand, we have the queen and the drones. They represent the reproductive part of the colony. They fly out to mate. During this, they search for so-called drone congregation areas, which are about 10 to 40 meters in the air. After the mating with 12 to 20 drones, the queen returns to her colony and begins to laying eggs, and that for the rest of her life. On the other hand, we have the sterile helpers. They are represented by the many worker bees in the colony. These take on variety of tasks in the hive, such as cell cleaning, brood care, queen retinue, and food exchange through trophallaxis. But food processing and wax production or processing are also part of the daily tasks. And of course, probably the best known behavior of worker bees, foraging. All these tasks are performed by worker bees at a specific age. It should be noted that these age limits are only guidelines. If necessary, nurse bees can, for example, engage in food processing. When we are looking at this division of labor, the question arises as how to such an organization is determined neuronally. To answer this or other similar questions, we must first ask another question. What are the possibilities for measuring neuronal activity? My presentation will be focused on answering this question. Let's take a look at the genetic toolbox of the honeybee. Firstly, we have the editing methods. These include methods where we can switch off or down regulate genes or even introduce DNA sequence into the genome. To date, the following three methods have been firmly established in the honeybee. Firstly, the injection of RNA interference, or RNAi for short. Here, the already transcribed RNA of the gene is destroyed by binding, preventing the translation into a protein. Additionally, there is the use of transposons. This protein can be used to introduce DNA fragments into the genome in an undirected manner. And finally, there's the CRISPR-Cas9 system. In other words, the possibility of targeted modifications in the genome and either switch off a gene or specifically introduce DNA fragments. In addition to these genome editing methods, we also find possibilities for visualization and analysis. Two standard methods that are already frequently used in the honeybee we are presented here. On the one hand, it is possible to mark genes with a fluorescent protein. This allows the precise localization of the area of gene expression. In addition to the fluorescent protein, it is also possible to introduce a calcium sensor into the genome. This can fluorescently mark active presynapses. Since we are interested in the question of how neuronal activity can be measured, this calcium sensor offers itself as a suitable method to answer this question. How exactly does it work? And how do we use it in the bee? Firstly, we need to modify the piggyback transposon system to carry the sequence for the calcium sensor. This is then injected into the bee egg, and we can raise transgenic honeybees. These are then prepared in vivo and stimulated with specific odors. The reaction to the odor can be recorded using a microscope. Let's start with the piggyback transposon system into which we integrate the calcium sensor. First, we need a so-called cascade. This cascade consists two main parts, a promoter and a coding sequence. In our experiments, we used a synapsine promoter. This promoter ensures that the substance gene is activated specifically in those cells that are responsible for transmission of nerve signals. The coding sequence that we insert into the cascade is derived from a fluorescent protein called GCAMP6F. This protein has a special property. It fluoresces when it comes into contact with the calcium ions. We then inserted this entire cascade into a so-called piggyback plasmid. This plasmid served as a transport vehicle with we can introduce the cascade into the bee's genome. The integration process works as follows. A special protein, the transposase, binds to the plasmid and recognizes specific DNA sequences in the bee's genome. At these sites, the transposase can cut the cascade out of the plasmid and insert it into the bee's genome. The cascade is integrated at the random location in the genome. Now the question naturally arises how this system can be injected into the bee egg. There is a possibility of confining a queen bee in smaller cages where the bag has replaceable honeybee imitations. This causes the queen can lay eggs on small brown plastic pieces with depressions. We can then inject the piggyback plasmid and the transposase mRNA using a binocular microscope. I've brought you a video for you to show what such an injection looked like. Here, blue food coloring was injection for illustration. With this system, we can inject up to 1,000 eggs a day. I can tell you, this is really hard. In our first experiment with the calcium sensor, we tested 16 olfactic odorants. These are different both in chain length and in the functional group. Since similar experiments had been conducted with these odorants before, these odorants served initially to control the system. Let's now turn the actual application of the system. First, the bee was immobilized on ice and fixed in such shoulders. Then we could remove the cuticle so that the brain was freely accessible. After 30 minutes, we could start the microscopy. First, 3 sections were recorded in which nothing happened. Then the odorant was presented for 1 second and the next 17 seconds were recorded as a reaction. I have brought you a video of such a recording. After about 3 seconds, the word odorant appears. At this moment, you can see very clearly a change in the recording. Since this may have been a bit fast, I would like to play the video again before I continue. What actually is happening in the cell during the recording? The bee is climbed in the holders and exposed to an odorant. This opens the calcium channels in all active neurons at the presynapse, allowing calcium ions to flow into the cell. In the cell, they are the inactive fluorescent proteins with the calcium binding site. As soon as calcium flows into the cell, it binds to the fluorescent proteins, which then begin to fluoresce. Before I show you the evaluation of an odorant presentation, I would like to give you a brief excursion into the topic of the honeybee brain anatomy. On the left, you can see a model of the brain. That should give you a raw overview. The optic lobes, shown here in yellowish, receive the visual input, while the antennal lobes, shown here in blue, receive the olfactory input. The grayish area, the lateral horn, all information is proceeded and forwarded. The mushroom bodies are shown in red. They are responsible for the storage of information. On the right, you can see a model of how the nerve pathways lead from the antennal lobes via the lateral horn to the mushroom bodies. This is the processing pathway of odorants in the brain of a honeybee. Here you can see the activity maps of the odorants 2-heptanone and heptanyl. Especially with 2-heptanone, you can see very well how all three areas involved in the olfactory processing are significantly more active than the other brain regions at this time. This system was thus the first system at this time that allows that the simultaneous recording of olfactory response in different brain structures in the honeybee. But let's take a look at the three areas one by one. Here you can see the activity maps of the antennal lobes for all 16 odorants. It is clearly visible that the presentation of each odorant triggers a signal in a different set of antennal lobe glomeruli. That means in a subunit of the lobes. But there are also similarity in the patterns. For example, here, here, or here. If we now relate the similarity of these patterns to the chemical characteristics of the odorant, we can find that both correspond to each other. Let's now take a brief look at the other two areas, the lateral horn and the mushroom body. More precisely, the calyx lip. Here too, we see a different processing pattern for all odorants used. I can tell you that that there is no longer any correlation between pattern and chemical characteristic here. A decoding of the odorant has already taken place in the way to the lateral horn. Nevertheless, one thing should be clearly mentioned here. It is now possible to measure neuronal activity at higher levels, such as the mushroom bodies. After I have now shown you the answer to some rather theoretical arguments, I don't want to withhold the reaction or measurability of the neuronal activity to bee pheromones. Let's start with two volatile odorants. On the one hand, ethylpentyl acetate, a component of the aggression pheromone, and on the other hand, beta-ocimene, which is secreted by young larvae. Here we see a clear signal compared to the control. The same applies to the non-volatile odorants. Here too, we see a clear signal for both the components of queen pheromones and brood pheromones. But why I've told you all this? How we can use the calcium sensor? There are two areas of applications. On the one hand, for research purposes, which corresponds to my area of application, and on the other hand, for the economy. In research, we can use the calcium sensor, for example, in the field of neuroethology, to uncover connections between behavior and neuronal activity with the help of pheromone use. And on the other hand, we have the economic benefits of the calcium sensor. Here, for example, the influence of pesticide on a bee colony could be tested. This would be of great benefit, especially in the field of agriculture or the approval of pesticides. With that, I'm almost at the end of my presentation, and I would like to briefly summarize the most important aspect of this genetic tool. The calcium sensor allows simultaneous recordings of different brain structures. This is useful for dissections yet undescribed sensory and/or behavior-related pathways, and allows recording of poor study structures, for example, the lateral horn or the mushroom bodies. This tool represents a major project for the neural ethology of social behavior. And of course, the economic use, the impact of pesticides. With that, I would like to thank you very much for your attention, and a special thanks goes to everyone who was involved in the establishment of the calcium sensor.